Hello, everyone. I guess it's time we start the webinar. I'll say hello and Happy New Year. Before I start, I'll just mention that there will be 5 minutes at the end when I will try to address questions that you can raise during my presentation in writing during the process of my talk. I'm Joël Doré, Research Director at INRA, based in Jouy-en-Josas in France, near Paris. And I'm also a scientific advisor to MaaT Pharma. My presentation is host-microbiome symbiosis in health and disease. And the slides are not moving. Okay, I'll start with this short page, disclaimer. I give lectures, do research funded by industry, editorial work, and I'm co-founder and scientific advisor to MaaT Pharma. And my learning objectives for this webinar are to give you an update on the human intestinal microbiome based on what we have learned from metagenomics, essentially to highlight symbiosis to dysbiosis transition in humans and to address implications for the future of nutrition and medicine. And why symbiosis? Well, we humans are essentially a symbiotic organism. We are born virtually sterile and we meet the microbial world and develop our microbiota at the same time as we mature our immune system. And our microbiota will contribute, as shown on the left here, trophic functions, metabolic contribution, barrier function, immune stimulation, and signaling to essentially every organ of the human body. And that leads to this unique recognition of the microbiota as part of self by the immune system, which is the key of our symbiosis. Maintenance of symbiosis is essential for health and well-being, but there could be disruptions at the level of ecology. This comes with the loss of function, essentially the barrier function, protection against environmental microbes, including pathogens, and disruption at the level of immunity, which is associated with risk of autoimmune or immune disorders. And so what we want to do is use any possible tool to understand symbiosis, to monitor symbiosis, and to fine-tune and correct or mitigate the risks. Now, we are humans, microbial, ecosystems, and symbiosis. And the figure here, this 100 trillion, is the number of bacteria that each of us interact with on a daily basis. We have microbes on the skin, we have microbes in the urogenital tract, microbes in the lungs, in the mouth and oral cavity, as well as obviously in the intestines, where the greatest population density and diversity is located. And we today count genes. So facing the 23,000 genes of the human genome, we have on average 600,000 microbial genes. So that leaves 4% for the contribution of genes of the human genome. And science of the microbiome has been evolving quite rapidly. So the landscape is what I will describe now. One element before I start, we know that by culture, we are able to only assess roughly 30% of the dominant intestinal microbiota. 30% of what we would see under the microscope if we were looking at a content, a human intestinal content. So this is very limited. So this is why we have developed tools, culture-independent approaches that allow to obtain a complete vision of the gut ecosystem. These 100 trillion bacteria per individual that are at the interface between food and the gut epithelium, in contact with the first pool of immune cells and the second pool of neural cells of the human body, hence now gaining recognition as a true organ, playing a major role in health and disease. So I will start with the metagenomic highlights. Now the metagenome is the name we have given to the combined genomes of all dominant microbes, in our case within the human intestinal tract. So the way we build knowledge on the metagenome is by extraction of total DNA from human intestinal contents, mainly stool samples, but we can work with biopsies as well. Apply whole genome shotgun sequencing, assemble and annotate genes to the best we can. And then the initial objectives of the European MetaHit program or the HMP in the US were to build the reference gene catalog for these ecosystems and to move towards rapid gene counting methodologies. So just in a few clicks, key lessons that we have learned. So we built the reference gene catalog, which highlighted core metagenome that I will illustrate, but also the presence of rare genes. We built tools, bioinformatics tools, to cluster co-abundant genes into genomes of metagenomic species. We also address questions in terms of ecological arrangements of the human intestinal microbiota, hence describing 3 ecological arrangements which we called enterotypes that I will describe. And we coined low gene count as a key stratifier in the human gut. And we applied metagenomics to describe the microbiota in several chronic disease conditions. Examples given here, type 2 diabetes, obesity, or liver cirrhosis. We've expanded that quite a lot, actually, to many other conditions, including neurological, neurodegenerative, or neuropsychological conditions. So the construction of the reference gene catalog led us to describe 3.3 million gut genes from 124 European subjects in 2010, and we expanded that knowledge until 2014, and this update that described 10 million independent gut genes. This is actually a major asset for the future of metagenomic profiling, and you will see how. Now, if you look at these curves, it's essentially the increasing number of genes that we observe from the number of subjects that we studied. We ended up with 1,267 European, Chinese, and American subjects. And this red curve at the bottom highlights the number of genes that are shared by more than 50% of the cohort. So it's essentially the most common genes, and they correspond to most common microorganisms. They're a small number that was captured after roughly 100 individuals. We are currently working on animals, pigs and chickens, and describing also massive diversity of genes in those ecosystems and corresponding massive numbers of species, 1,500 for the human, up to 2,300 for the chicken, actually. I mentioned the development we made into describing genomes. So what we do in this case is we base our calculations on the fact that all genes of a species will have the same abundance in an individual. And so just clustering genes on the basis of co-variation allows to reconstruct pools of genes that will correspond to genomes. So we described in 2014 741 what we call large metagenomic units of more than 700 genes. We're able to demonstrate that they actually correspond to bacterial genomes, just recognizing known bacterial genomes, in spite of the fact that 85% of the genomes we describe in doing so correspond to previously unknown and yet non-cultured microorganisms, bacteria that are not represented in culture collections, for example. There were also in this work a lot of small metagenomic units corresponding to phages, plasmids, or CRISPR elements that are most commonly associated to one single bacterial genome or microbial genome. So the pipeline that we apply today is illustrated here. It starts with recommendations for sample collection and clinical trials, for example, provision of kits for collection, total DNA extraction, which is now in the process of automation, high throughput sequencing, generating millions of small sequence tags that are not used any longer to reconstruct genes, but that are used to map on the reference gene catalog. And the mapping allows to count genes and to generate gene abundance profiles that are roughly immediately translated into metagenomic species abundance profiles. And it's only in the end when those are integrated together with human data, with clinical data, with a response to treatments or diet, for example, that we identify relevant microbial players and build prediction models. The illustration given here is sort of a barcode that illustrates the occurrence of genes in each and every individual as columns, and rows correspond to bacterial species. Standardization is really critical in the process, and I indicated here the website microbiome-standards that we use to post standard operating procedures that were eventually published in Nature Microbiology in 2017. In Nature Biotech, I think there's a mistake here. Okay, so this is the consortium that was gathered for the work on microbiome standards. IHMS stands for International Human Microbiome Standards, and we analyzed the protocols implemented by 21 different laboratories for DNA extraction and came up with only 2 procedures that were recognized as convenient for the future work. When we look at a large number of individuals, what we see is a fairly limited number of species that are highly conserved. Present in 90% of subjects in a cohort of 124, we have 57 species. If we increase this occurrence percentage or prevalence, only 18 species are found in nearly each and every individual. They are listed here. They correspond to the major phyla that we do observe in the human gut microbiota. It's less than 20 out of, on average, 200 dominant species per individual. So that means that there will be a massive inter-individual variation. I mentioned we looked at ecological arrangements, and this illustration published last year in 2018 illustrates what we observe when we look at a large human population. We essentially see 3 different ecological settings. When we started this exploration, we expected to find the average human gut microbiota. And in fact, what we observe is this distribution in 3 different ecological arrangements whereby bacterial species and genera will essentially arrange in closed settings of microbes. Now, as the illustration shows, it's not a fixed situation, so there can be flexibility or plasticity in the ecological distribution, and one person may actually move from one enterotype to the other during life, even within 1 year, with change in diet, for example, or antibiotic treatment. In just 1 year after the first description, Gary Wu in the US indicated that dietary habits have an impact on the belonging to enterotypes. And essentially, Bacteroides enterotype is dominant among individuals that have dietary habits that correspond to what is commonly called the Western diet, essentially fast food type. Whereas Prevotella essentially is highly associated with a high fiber diet, more fruits and vegetables dominated diets. And we, in this work, were able to identify that there was a connection with gene count or richness, and we studied this parameter much further, showing that when we look at the distribution of the human population in number of individuals as a function of number of genes in the human metagenome, ranging from 200,000 to more than 800,000, we did not have a regular bell-shaped curve, but rather shoulder to the left corresponding to low richness microbiota individuals, and a main peak to the right with high richness microbiomes. What is shown in brown here is the enterotype. So you see that the Bacteroides enterotype is dominant on the left-hand side, whereas the 2 others are more represented on the high richness microbiota. There were 68 species that were significantly linked to this parameter, and we were also able to show that later on, low gene richness is a key stratifier. corresponding to, associated with worse phenotypes, non-response to treatment, to diet, or higher risk of comorbidities, especially in metabolic disorders. So to conclude on this part, this metagenomic view of the fecal microbiota showed that each dominant microbiota gathers on average 600,000 genes, but they actually represent a high diversity within the human population. Roughly 200 bacterial species, 25-fold the size of the human genome. We built a reference catalog of some 10 million genes. This is a true asset for the future of analysis. We have genomes of yet uncultured metagenomic species that can be assembled. The microbiota can be characterized by quantitative metagenomics. Small proportion of the diversity of genus species constitute a shared metagenomic core, probably contributing in a major way to functionalities of the gut microbiota, and individuals will vary by gene richness, metagenomic species, and ecology or enterotypes. Now I have a segment on symbiosis per se. And I will start with this observation of epidemiology. Essentially, in spite of considerable progress in medicine that through the second half of the previous century led to this observation of a ever better control of infectious conditions, what was observed is this rise in chronic immune-related disorders, including inflammatory bowel disease. Crohn's disease is shown here. Ulcerative colitis would be on the same trend. Autoimmune disorders like multiple sclerosis or type 1 diabetes, allergies, but you would have on the same curve metabolic conditions like type 2 diabetes or obesity, liver conditions, NAFLD-NASH, for example, cirrhosis, and some cancers, as well as neurological conditions. This is leading us to 1 human in 4 concerned by any of those chronic conditions by 2025. So prevention is an urgent need, and it's leading to think that human longevity might actually be at stake if we pursue with this uncontrolled rise in incidence. Just as an example, incidence of autism in the USA is such that 1 birth in 59 is bound to be concerned during its life by autism, and it's 1 out of 150 in the UK or France today. So we can, it's justified to question what we have neglected, and just a few examples of what maybe will have impacted the microbiota among things that we have drastically changed over just a few generations. Birth mode and environment, we have essentially changed everything. And if I just want to highlight 2 elements, C-section birth is today the case for 90% of births in several places on Earth. And this is preventing natural vertical transmission of microbes from the mother to the child. The resource of antibiotics at birth is also quite common. In the US, it's commonly 2 treatments at birth on a preventive mode. We have also changed nutrition and physical activity, but when we talk of nutrition, we think of the contribution of plant material to energy. It commonly was 60% at the beginning of the previous century of the energy. It's down to roughly 10% today. In terms of grams of fibers per day, it's 17 grams in France, when the nutritional recommendation is more like 25 to 30. And we also have exposure to xenobiotics that we know is impacting the microbiota from just a few publications that indicate that it's tentatively pro-inflammatory when we think of, for example, emulsifiers in food that we are exposed to every day by processed food. And this neglecting the fact that we are microbial. We are this 1 kilogram of bacterial biomass or more. 50% of our cells are bacteria, and 70% of this is unknown by culture. So I mentioned this is led to the description of a connection between microbiota and alteration and disease conditions, but there's beyond microbiota a common thread in chronic conditions that concerns as well alteration of gut permeability, alteration of the inflammatory state, and coming with those oxidative stress. So essentially what we have is not just a microbial story, a story of the relation between the host and its microbes, and the story of symbiosis. One of the ways to depict this is presented here. So when everything is going well, when we have symbiosis, we have a rich, diverse microbiota, we have integrity of the mucosal barrier, immune tolerance, and a balanced redox state. And when there are alterations, what we do see most commonly is loss of richness of the microbiota, loss of symbionts, increase in pathobionts, essentially gram-negative pro-inflammatory microbes. And together with this, we observe intestinal permeability to hyperpermeability. We observe a low-grade to overt inflammation and oxidative stress that will further aggravate the alteration of the gut microbiota. So essentially, we can establish a vicious circle that will explain this durable alteration of symbiosis, following what Martin Scheffer calls a critical transition. So when we would like to be in this reassuring state of a continuum between health and disease, if we indeed have a vicious circle, then actually what ecological theory tells us is we have disruption, we have complete what is called critical transition between one state and another. And well, it's not done today, but essentially there would be 4 targets corresponding to the 4 triggers of the vicious circle that could be applied to diagnosis, to prediction, to prevention, and to therapy. Hopefully this will be available for the future. So just to give an illustration of critical transition and alternative stable states based on the writings of René Thom 1983 and Martin Scheffer more recently in Holland, as long as the stress conditions applied to the ecosystem are reasonable, then we can actually have the ecosystem shifting but remaining within its initial stable state, healthy. And then if we push the stress further, then we will bounce in a state of equilibrium that will be typical of pre-disease or disease. And even if we remove the stress condition, essentially once it has bounced with this vicious circle activation, then it will remain for a long time in the altered condition. Only if we drop stress conditions very, very low can we hope to be able to reinstall the initial normal condition. I will finish with the third segment on microbiome-based innovations. What can we expect from microbiome discoveries and description? We described in 2017 in a paper published in Therapy, after work with quite a large number of pharma industries, these 4 segments of applications. I will essentially today discuss stratification and monitoring, how the microbiome data can serve this aspect, and how microbiome can be used as essentially a drug of its own. So you will all have seen that previously. It's work by Eric Palmer that concerns the microbiome assessment of patients receiving hematopoietic stem cell transplantation. At the moment of HSCT, patients were divided into high diversity, intermediate diversity, low diversity, which is illustrated by the number of colors, essentially microbial taxa, shown in these graphs. And the fate of the patient is followed over 3 years thereafter. And it was essentially shown that there's a connection between the richness of the microbiota and the prognosis for the patient. It's summarized here. Essentially, patients with high diversity microbiome have a probability of transplant-related death, which is fairly low. This is around 11% here. Whereas at the other end, patients with low diversity microbiome, inverse Simpson index below 2, have probability of transplant-related death greater than 50%. Again, intestinal microbiota richness last year was shown as a predictor of response to anti-PD-1 anti-cancer immunotherapies in the work of Laurence Diller and colleagues in the case of non-small cell lung cancers, showing that metagenomic species count is higher in long progression-free survivor patients. Whereas in the work of Jennifer Wargo, it's the case of melanoma patients, and what was shown is that alpha diversity of the gut microbiome is higher in responders to anti-PD-1 than compared to non-responders. Now, we had worked with my colleague Patricia Lepage and Laurence Diller before that, showing that gram-positive symbionts, mainly gram-positive, can act as adjuvants in anti-cancer therapy. In this case, it was worked with chemotherapy cyclophosphamide that was leading to the concept that gut permeability induced by the treatment will allow translocation of bacteria, and some bacteria will actually in lymphoid organ prime a Th1, Th17 response that in tumor beds will favor the efficacy of the treatment. And in this paper and following papers in Science, it was illustrated that the fact of using antibiotics could actually knock down the microbes that are the ones that are adjuvant, promoting the efficacy of anti-cancer therapy. And that leads to illustrate in mice that receive a tumor will actually lose the efficacy of the treatment if they receive antibiotics at the same time. I will get back to this with human data. Now, looking in greater details, Jennifer Wargo described that in the efficacy of anti-PD-1 in the melanoma patients, a longer progression-free survival is observed in patients that have a higher abundance of this bacterial taxon Faecalibacterium. And it's illustrated here. It's highly significant after 600 days. And conversely, there was a connection between shorter progression-free survival and the abundance of Bacteroidales, the order Bacteroidales, which is the dominant gram-negatives within the human intestinal microbiota. Now, I was involved in description of Faecalibacterium a long time ago, but ever since we have been working on this bacterial taxon, especially the species Faecalibacterium prausnitzii, which is a gram-positive, a major contributor to the production of butyrate in the intestine. It was connected with inflammatory bowel disease, but not only, also colorectal cancer, IBS, a few other conditions. We were able to show that it expresses anti-inflammatory activities, and it's even in Crohn patients that are set in remission, that are set in quiescent phase of the disease. It's a predictor of remission. The figure that came out of this work is interestingly quite similar to the one we have seen from the work on the melanoma patients. So Faecalibacterium abundance in itself is one of the strongest predictors of response to anti-PD-1 in melanoma patients. Now, antibiotics again in humans this time, based on work in non-small cell lung cancer patients, renal cell cancer patients, urothelial carcinoma. What was shown is that if the patient received antibiotics, and nearly any antibiotic in the 2 months that precede or the months that follows the initiation of immunotherapy, then their overall survival was shorter. It's illustrated in the red curve compared to the black curve here. So I will finish with an illustration of bacteriotherapy or microbiotherapy. In this slide, what I'm showing is specific bacterial species that have been described for their bioactivity, essentially protective bioactivity. They are many members of the metagenomic core of the microbiota. Some of those microbes have led to the description of a specific bioactive molecule that is expressed very often anti-inflammatory, for example. And in some cases, this has led to the inception of private companies that are today preparing clinical trials, essentially administering those microbes into capsules to patients in order to treat or mitigate risks in specific disease conditions. Now, this is the one single bacterium approach. You will have heard of fecal microbiota transfer as another approach that is developing in many conditions. It is a recognized treatment today by regulatory agencies for one single indication, which is recurrent Clostridium difficile infections. And indeed, in the work of Van Noot, it was shown that when the last resource antibiotic will essentially eradicate the pathogen in 30% of the patients, fecal microbiota transfer, one round or possibly two will cure the pathogen in more than 90% of the patients. And so that has in fact led to the interruption of the study at intermediate analysis, considering it was non-ethical to pursue with such a higher efficacy of FMT in this case, and to the acknowledgement of this treatment second line in recurrent Clostridium difficile infection today. Now, this approach is explored in many other conditions, and I will just give one illustration from a proof of concept study that was implemented by MaaT Pharma in patients that hematologists will obviously know. It concerns a trial in AML, a phase 1b/2a trial, 25 patients that were administered a single dose of autologous MaaT Pharma microbiome restoration biotherapy. Indeed, the patients were sampled, so one stool was collected at diagnosis before induction chemotherapy and antibiotic treatments, and then following this 1 month, patient received, after another collection, their own microbiome. They were back in the hospital 10 days later for consolidation chemotherapy and followed over time for 1 year. What was observed is that at the level of the overall microbiota, there was a marked disruption of the microbiota induced by chemotherapy followed by antibiotics. But after autologous readministration, the microbiome was restored quite significantly. Looking specifically at bacteria that will contribute beneficially to the symbiosis, it was shown that they were essentially dropped to very, very low level following chemotherapy and antibiotic treatment and restored completely following autologous transplantation. And neopterin as a marker of inflammation was indicative of an inflammatory burst following chemotherapy and antibiotics. And again, the system was reset to a homeostasis after the treatment. 90% of the microbiota was recovered by MaaT Pharma microbiome restoration. And it was even shown that there is an excellent safety profile and 84% survival after 1 year, when the expectation on the basis of historical data is around 70%. And incidentally, MaaT biotherapeutics are available today for clinical trials and compassionate use. So I will summarize. Humans share a core microbiome, and yet they differ by genes, by species, by enterotypes or ecology, and gene count. Microbiome gene count is a key stratifier of health, essentially, and this is true in several immune disorders and clinical conditions. Dysbiosis is an altered state of host microbe symbiosis, and it's observed essentially in the context where there can be auto-aggravating loops or signals from both sides, the host and the microbiota. Microbiota modulation should be considered as a target for personalized nutrition as well as therapy, and nutritional bioactives, but also live microbes can be considered strategic bioactives for the maintenance, the preservation, or the restoration of host microbes symbiosis. And obviously, I'm a microbial ecologist, but I like to say that a major way of leveraging prevention will hence be related to the management and the monitoring of intestinal ecology. I will thank you for your attention, and hope I have a few questions to address that should pop up on my phone in the coming minutes. So if I have the organizers, I don't see. Oh, here come question, not yet. Okay, so I welcome any question at this stage. So I guess there are no questions coming. If you are keen on doing so, it's also fine for you to send me questions by email. My email is joel.dore, joel.dore at INRA.fr. Thank you very much.